congratulate all of you for sticking out to the almost second to the last talk. As was mentioned, so the title of my presentation is Laboratory Science to Support Risk-Based Quality Assessments. And this whole concept of quality assessments, I hope to show you, and this is something that our OPQ director, Dr. Kapcha, mentioned yesterday, is really how review and laboratory divisions, as well as other divisions, are all impacting the materials submitted to the agency. I am actually based out of St. Louis, but half of our office in St. Louis, half of our office is here at the White Oak facility down the street. But I'm really happy to be representing OTR up here, and thank the staff for the invitation. So OTR has a science and research program, and the science program is aimed primarily at looking at problems that are at hand in front of us right now, particularly consumer complaints and public health issues. I know this was covered in some of the presentations yesterday afternoon, where patients contact the agency. Maybe they claim that a product is not working. Maybe they were taking the brand product. The doctor or the pharmacy switched to the generic brand. It's not working the same. And I think that all of that, once it comes to the agency, sometimes then a laboratory consult is requested. And quite often we're the lab that is charged to look at that. Some of the work also goes to our ORA labs that are spread throughout the country. But our lab in OTR does handle, maybe about 10 or so of these cases a year. Also, we look at public health issues. We concentrate on rapid response and really take pride in trying to get to the problem and find a solution and communicate to our stakeholders in a rapid fashion. The research program is a little bit more forward-looking. So that is aimed at looking at challenges that maybe not in front of us right now, may come down the road tomorrow. And it's very important that we maintain a state of preparedness for such projects going forward. And so OTR and OPQ is really invested in looking at new and emerging technologies, developing advanced analytics, and particularly for trying to stay current with the science to tackle the issues of complex drugs. So in generic drug science, OTR and OPQ work hand in hand with our Office of Generic Drug counterparts. Some of the work that we do as far as laboratory consults goes, method evaluation, we call that method verification. It's a little different from traditional method validation that you may be familiar with. And really, oftentimes, this encompasses maybe a targeted look at some aspects of the analytical package that is submitted to the agency. It's kind of the miracle happens that Bob mentioned in his presentation earlier today. Sometimes we're asked to find out, how did that miracle happen? And so we're looking at certain aspects. And I will just mention that's why also it's very important to provide detailed methods for us to look at as well. We also, our laboratory consults are focused on product quality and pharmaceutical equivalence. And oftentimes the review divisions ask OTR to look at maybe side-by-side comparisons of brand and generic drugs. OTR is also involved in the guidance and standard development, particularly in providing laboratory data. A lot of the guidances that we see on the table outside for those here in the room, for many of those, OTR was requested maybe to provide a lab analyst, a subject matter expert, or maybe to provide some preliminary data as well. And also developing improved testing protocols. So OTR does serve on several of the expert panels, working groups, through our compendial operations in OPQ. And finally, and this really, I think, is where the whole quality assessment concept comes into play, because we perform research, we perform science in the lab, but one of the big aspects and one of the most impactful things that we do as well is to exchange that information with the review divisions. And OTR holds a few, maybe about 5 to 10 workshops with the review divisions per year. Some of it's here in Maryland, some of it's in St. Louis, where we give reviewers the opportunity to do science firsthand with on-the-job, hands-on training. These workshops are about 1 or 2 days, and so it provides, kind of a continuing and modernizing aspect to the knowledge base and the review staff as well. And we get feedback, and we really do appreciate that feedback when we do that, because then from a lab perspective, we can better serve our review counterparts' needs. So I'm going to give you really 4 case studies here today. A lot of these are going to be just pretty quick overviews. Many of these could probably be 20-minute presentations themselves, but I do encourage you that the slides will be available, and certainly, a quick Google search of some of these will get you the paper that's associated with many of these issues. So the first one is a consumer complaints, maybe quality issues in transdermal systems. Secondly, we'll move on to enteral feeding tubes, and particularly some of the aspects that are observed in clinical settings. We'll also look at emerging tools, particle profiling, and nasal spray drugs. And finally, as part of the research program, trying to move more towards biorelevant dissolution methods and looking at the effects of contraction on drug release. So for the first case study, it's on transdermal drug delivery systems, and I thought that Dr. Chasen yesterday mentioned and really covered a very interesting and very, really, it crystallized just how real the problem is in his patch example, where the generic drug patch, I think, was maybe 2 or 3 times the surface area of the brand name drug. And so transdermal drug delivery systems come in all different shapes and sizes. And one of the areas that's often something that we look at is the release liner. And the release liner can come in the same size as the transdermal system itself or be larger. And you see the array of different examples here in the slide. So with release liner studies, OTR has responded to some consumer complaints where the patch won't heal cleanly, or maybe the medication is sticking to the plastic. These are comments that we've had, and we actually perform laboratory studies. We have devices, some are custom, that we have built over the last 15 or 20 years that the St. Louis lab has been able to develop that methodology. Also, we perform consults from the review divisions. Sometimes we're looking at side-by-side comparisons, interlaminal adhesion studies between the brand and generic formulations. And we're very happy when we hear from the review staff that these studies that we perform in OTR maybe impact maybe some of the communications that the review divisions have with the firms. One of the areas in transdermal drug delivery systems that is our forward-looking area where we're trying to develop enhanced analytical toolbox is in the crystal formation in transdermal systems. So crystals, especially those that the consumer may be able to visually see, which does happen, can be very alarming. And from a quality perspective, this may cause skin irritation, loss of adhesion, reduction in the drug delivery, and maybe an uneven appearance. And so we have seen aspects where we are able to see crystals visibly. And from a laboratory perspective, one of the things that we try to do is try to develop a technique, and here is a scanning electron microscopy, SEM Raman technique, where we can look at both the image of the crystals and also the chemical fingerprint, which is shown at the bottom panel simultaneously. In the second case study, this is enteral delivery of proton pump inhibitors. Certainly, we probably all have some experience or know somebody that may take these medications daily because it is one of the most widely prescribed drugs in the US. And primarily, it's used to manage acid-related disorders. So we see an example here of the brand name drug, Nexium, on one side. And we also see one of the patients, a pediatric patient, that has been fitted with an enteral feeding tube. One of the areas that the enteral feeding tubes are used to deliver these type of drugs are for patients such as this infant that we see here that have difficulty swallowing. And so what happens is the granules are emptied into a syringe and then delivered into the tube. And what's unique about this specific case, which we have several papers cited on here, including a paper of our own in the next few slides, is that the granules inside the capsule are enteric coated. And so one of the things that we did a laboratory investigation on, what happens with the enteric coating as a result of the clinical preparation, okay? So with enteral feeding tubes, there are several really key risk factors to consider. And it's more than you think, because in addition to the size, composition, and geometry of the tube, several examples are shown at the bottom of the slide, you also have to consider the syringe used to deliver, and also such innocent or mundane things as the water source. Everybody here, if you come from a different part of the country or from the world, listening on the Adobe Connect, every water source is a little bit different pH, different profiles, and so that plays a role in the whole process. So in the laboratory, what we did is we performed an acid resistance stability study, and we looked at 4 conditions. We looked at DI water, tap water, and DI water and tap water with 15-minute incubation times. And we did that because we wanted to see what is the impact of preparing the syringe and delivering immediately versus preparing the syringe and maybe having a hold time. And so in this plot on the vertical axis is the percent API dissolved, and then we looked at 5 different lots. And all lots passed the acid resistance test when delivered immediately. And you can see those are the small bars that are on the leading edge of all those 5 groups. But 4 out of 6 lots had significant amounts of API released when the 15-minute incubation time was taken into account. And that indicates that the enteric coating is breaking down in the syringe. That may lead to undesired effects, maybe efficacy effects once the product is delivered, if that enteric coating is not able to remain intact. The third case study is something that OTR has been looking at for at least 15, maybe even 20 years, longer than the time that I've been there. So is nasal sprays. And we do have a research program that has looks at inhalation products. And particularly with nasal sprays, one of the critical quality attributes is particle size. So particle size is well known to impact effectiveness and bioequivalence, but at the same time, particle size is not necessarily easily characterized in some of these nasal spray products. Primarily, the active and inactive ingredients coexist in solution, and as was mentioned in several talks too, polymorphic forms and transformations may also play a role. And so one of the tools that we have been working with OGD in collaboration for a few years now is looking at how a automated system, in this case the morphology-directed Raman spectroscopy system, can help perform that characterization. So this is a very interesting system. We have this instrument in St. Louis. And it's interesting because once you get the experiment running, it's a set it and forget it technique. The problem is building up the expertise to get it running and then looking at the data analysis once you have 8 hours worth of data to look at. The morphology screening portion of this test is used as a classification. So you can use any number of parameters, convexity, circularity, contrast ratio to filter out the particles And then once you determine how you're going to filter out the particles, because you may find thousands. We're talking about 10, 50,000 particles. Then you can request the automated system to look at and perform chemical identification of these particles. And in this slide, we see 3 different chemical fingerprints. We see the excipient in the blue panel at the top, and we see 2 different polymorphic forms. Now, to the naked eye, I know there may not be a lot of spectroscopists in this room or in our virtual session. They may look similar, the second and third panels. But one of the areas that we're able to see differences is circled there, where the different polymorphic forms have a unique characteristic. And this is all something that we've been able to look at formulated products to identify different polymorphic forms, and been able to also, I know industry uses this, and the FDA has been able to use this as well in some of the consults that come into our lab. And finally, this is the last case study. This is with Nifedipine extended release tablet dissolution, another area where we have collaborated with the Office of Generic Drugs for a number of years. So here we have 2 different products. We have the brand name product, product A, which is an osmotic pump delivery system, and a generic product, product B, which uses a polymer matrix formulation. You can see there the before and after pictures of the product, and really how product A really remains unchanged as a result of the dissolution run. But polymer B, visually, you can see the polymer has expanded, and you know, in appearance, it's much different. And we looked at the effect of compression, so trying to model GI contraction forces on the 2 products, and we see that in the panel to the right, actually, so product A. It's tricky to being up here saying right and left, so I'm not even going to try doing that. So in the product A panel, you see that whether the contraction force that is delivered is 0.1 grams or 400 grams, there's not a huge difference between the dissolution profile. Now, in the case of product B, you see that the 0.1 gram and the 400 gram compression forces lead to vastly different release profiles, with the blue line, the blue trace in the product B panel, to be twice as much as the green line, the green dissolution profile. And so what that's indicating to us is for these 2 products, there's a GI contraction dose dumping observed. And this is important information, especially when we're trying to compare generic and brand name products. So I think I'm good on time. But like as I mentioned, a lot of these, I went pretty fast, but you'll be able to look at the slides, and if you have any information, I don't think it's too hard to find us for questions. But I really do hope that these 4 case studies show how generic drug science is very important to OTR's work and OPQ and even OGD's work, and we take that very seriously. And it's very important for science and research aspects to be part of our laboratory science, because we're not able only to respond to the project in front of us, but also be proactive in being ready for what's coming tomorrow. That's not always on the radar. And really, I think that I do appreciate your time. I think there's going to be, well, so here are some of the scientists that were involved in each of these aspects. That's our lab in St. Louis, but I really do appreciate your time.